Library for Human Genome Research, National Autonomous University of Mexico, and an international fellow at the Wellcome Cancer Institute in UK. She studied a Bachelor in Science in Genome Science at UNAM and a PhD in cancer genetics at the University of Cambridge. Her research focuses on using large-scale sequencing data for investigating the driver alterations, risk factors, and potential therapeutic targets of types of cancers important in Mexico and Latin America, such as acral, melanoma, and hepatocellular carcinoma. Please welcome our speaker.
Hi. Just making sure everyone can hear and see the presentation. Yes? Okay, perfect. Okay, so it's really a pleasure for me to be here with you guys. I'm really sorry that I cannot be there in person, but we are generally quite close. Here our lab is in Querétaro as well, in Juriquilla, in the Juriquilla campus of UNAM. And it's really, you know, a very happy occasion for me to talk to you because in our lab we've had two students from ASU, Jackie Romani and Sebas, who have worked with us, you know, in different projects that I'm going to touch on during this talk. So it's just great to talk to you, and thanks so much to the organizers for the invitation to speak. Okay, so maybe switching gears a little bit, we're going to talk now about cancer. And I want to tell you what we do in our lab to study cancer, study the causes of cancer, for example, try to understand how it evolves and hopefully how to attack it. And one of the most important focuses of our lab is on Latin American patients. We know that most of our knowledge is derived from studies in people that are of European descent, usually people that live, well, in Europe, for example, in Australia or in the US and Canada. And there's really a lack of diversity in the studies of populations, specifically in cancer genomics. So these are some of the topics that our work tries to address. And here in this talk, I'd like to tell you a little bit more about what we're doing. And of course, if you don't understand something, very happy to answer questions at the end. I understand that you're getting talks on many different topics this week, so I know that it may be hard to just grasp all the details. But okay, this is the title of the talk, Genomic and Transcriptomic Profile of Melanoma in Latin American Patients. And what I mean by genome and transcriptome is basically, genome is what the DNA of the cells are, and we're interested in the DNA of cancer cells. And transcriptome refers to what genes they are expressing, these cells, right? And many cells have many different transcriptomic profiles, which give them their identity, right? So just to keep in mind while we talk. Okay. So this is the outline of this talk. First, I'd like to tell you a little bit about this problem, this type of cancer in Latin America, melanoma. Then I'll tell you a little bit about what we're doing in our team. That includes findings from doing sequencing. I'll tell you a little bit more about that, of these tumors in Mexican patients, and also how we're building tumor models from Latin American patients to study these cancers more deeply. And then finally, a take-home message and some acknowledgements. Okay, let's start with an introduction. So basically, let's talk about what is melanoma. So melanoma is a type of cancer, and it is the most aggressive type of skin cancer. There are many different types of skin cancer. For example, you have basal cell carcinomas or squamous cell carcinomas. But you know, of all the different types of skin cancer, melanoma is the most dangerous. And that is because even though it constitutes a very small proportion of the skin cancers, only about 4%, it causes the majority of deaths from skin cancer, about 75%. Now, what is melanoma really? Well, the name comes because its cell of origin is a melanocyte. So a melanocyte is a cell. If you see here in this graph, you have just a graph of this diagram of the skin, and you see three layers: the epidermis at the top, the dermis, and the hypodermis. And if you do a zoom in here in the frontier, let's say before the epidermis and the dermis, then you see that the melanocytes live here in what is called the basal cell of the epidermis. And their job really is to protect our skin against damage caused by UV, basically sun exposure or exposure that you get from these sunbeds that cause a lot of damage to the skin. So what happens is, if melanocytes sense that that UV light is incoming and hitting the skin, they produce this a pigment called melanin, and then they distribute it to all these cells that they're in contact with through these little dendrites that they have. And this melanin is able to absorb the most damaging components of UV light. And in this way, it protects DNA from being damaged by UV light, because UV light, if it gets to the DNA, can cause mutations. And mutations, if they accumulate, they cause cancer. They can cause cancer. Despite this, however, the majority of the cases of melanoma are caused by UV radiation. So about 90% of melanomas can trace their origin to some event of sun exposure, for example, acute sun exposure. People in the UK that have lighter skin, for example, and go out frequently to holidays where they just lie down in the beach, for example, and get a lot of sun, those people are at higher risk of melanoma. And about, as I was saying, 90% of the cases are actually caused by UV light. Another interesting thing about melanoma, and the reason why we study it, is because the incidence of melanoma has been increasing in the last 40 years. This graph that you see here is from the US, where you can see that the lifetime risk of developing melanoma in the 30s and 50s was very low, but recently it has multiplied about fourfold. And this is not only the case in the US, it is the case in many places around the world. For example, here in Mexico, the National Cancer Institute of Mexico estimates that the incidence of melanoma has increased about 500% in the last 40 years. So this is a problem worldwide. And people speculate, there's a couple of reasons why this may be. The first one is that holidays have just become much more popular in the last few years. It is cheaper and accessible to basically take a flight to a beach and then just like get a lot of sunlight with no protection. And that, as we have discussed, increases vastly the risk of melanoma. That is one reason. And the other reason is that we have become now much better at detecting melanoma with new technologies and new devices that are much better at detecting melanoma in its earliest stages. So that also makes the numbers of melanoma increase, that I diagnose. So a combination of these two factors is why we have this big jump here in the incidence of melanoma. And finally, another interesting thing is that it is highly refractive to chemotherapy. So if it's detected in its advanced stages, melanoma almost does not respond to chemotherapy. And this is important because in countries like Mexico and many places, for example, in Latin America, the majority of patients are still treated with chemotherapies. This may be also because chemotherapy works, many forms of chemotherapy work by inducing a lot of DNA damage. And if cells get a lot of DNA damage, then they cannot survive and then they die. So, it is the principle that if chemotherapy comes and causes more mutations to cancer cells, they will die, which, in some cases they do. But in the case of melanoma, that is highly resistant to UV light and accumulates loads of mutations without dying, then in many cases, this is not the case. So these are some of the most important reasons as to why we study melanoma, and we think it's an interesting problem. Finally, if you look at the genome, and this will become interesting later, more than 85% of what we call cutaneous melanomas, I'll tell you about this distinction later, have what we call driver mutations in these three genes. A driver mutation is defined as a mutation that helps the cancer grow. And that's why you see it in a lot of these tumors, because if they, by some lucky event, get one of these mutations, then that cell can grow a lot and form a tumor. So let's say it's selected positively, having these mutations. And these three genes work in the same biological pathway, which some of you might have already heard about. Is called the MAP kinase pathway. And this pathway is important for cells because it's a growth pathway. So just to say then, to summarize, that more than 85% of cutaneous melanomas have mutations in any of these three genes, that their result is to increase the activity of this biological pathway, which results in cell growth. Okay, I hope this has not been a lot of information very quickly, but it will become important as we go through this presentation. Okay, but in the introduction and in the program, you have a word, acral melanoma. So what is acral melanoma? Okay, so like many types of cancer, melanomas can be subdivided in different histopathological subtypes. And here in this slide, you're seeing the 6 major histopathological subtypes. The first 3 are the what we call the cutaneous melanomas that I mentioned in my last slide. These 3 are, their origin is mostly due to UV exposure. And for example, superficial spreading, as its name says, is basically a tumor that expands first horizontally in the skin and then invades. And it is the most common in people of European descent. Then nodular melanomas, as you can see in this picture, grow very quickly vertically and invade much more quickly the deeper layers of the skin. And they have a worse prognosis than people that have superficial spreading melanoma. Lentigo malignant melanoma are melanomas that appear usually in sun-exposed sites in older individuals, generally, for example, in the face. And as I was saying, these 3 are related to UV exposure. Now, the 3 on your right are much rarer types of melanoma, and they do not have to do with UV exposure. The first one that you can see here is mucosal melanoma. They appear in mucosal sites of the body. For example, it can be the mouth, the sinuses, the vagina, any place that has mucosas is capable of developing mucosal melanoma. Uveal melanoma, which arises in the eye. And finally, acral melanoma, which is the focus of this presentation. So let's focus on this subtype from now on. What is acral melanoma? So this type of melanoma arises in the palms of the hands and on the soles of the feet and subungual locations, which means under the nails. Because of how it looks like, it's usually misdiagnosed as other conditions. For example, here in Mexico, it's much more common to have other conditions like diabetes or ulcers or fungal infections. And because these lesions can look very similar, they're usually misdiagnosed for these much more common conditions. And that means also that when people are finally diagnosed as having cancer, this is usually quite late, and you will see this in the stats of the tumors that we collect for this study. And finally, it is a rare subtype of melanoma, as I was saying, as it comprises only 1 to 8% of diagnosis in European descent individuals. However, if you look at a map of the distribution of melanoma subtypes in different countries around the world, then we see that actually, as I was saying, superficial spreading is the most common in green in many countries like the US, the UK, or Australia. But if you look at countries such as Mexico or Peru or some in Africa and Asia, then you see that from the total of melanomas that are diagnosed, acral melanoma is actually the one that constitutes the most number of these melanomas. So we can conclude that this type of melanoma, even though it's considered rare, is still the most common type of this disease in several countries in Latin America and Africa and Asia. And also, not only that, but it is also understudied. If you go to the National Library of Medicine, where there are the clinical trials for new medicines registered, and you look for acral melanoma, then you see that there's only about 41 studies out of more than 3,000 for all melanomas. So as you can see, it's very few. And also, not only is acral melanoma understudied, but also the populations where it is more common. For example, Hispanic populations or African populations are understudied as well. And what you're seeing here in this graph is the collection of samples that are in different repositories, in cancer repositories. This first one is called the Pan-Cancer Analysis of Whole Genomes. The second one is the Cancer Genome Atlas. The third one is COSMIC, which is the Catalog of Somatic Mutations in Cancer. And finally, the National Cancer Institute Patient-Derived Model Repositories. And these are all focused on cancer. And if you look at the samples and the patients that are actually part of these studies, then you see there's a lot of blue, which means there's a lot of European patients. So we have a problem here that is not only is this disease understudied, also our populations are understudied. And these are some of the most important motivations for us to do our work. Okay, so just before I go into the actual work, I also want to tell you a little bit about doing genomic research in Latin America and other low and middle income countries. It is sometimes considered to be harder to do genomic research in these countries for a number of reasons. First is, as I showed you in the last slide, that there are not a lot of studies of our populations available, which means it's harder sometimes to compare your results to an equivalent population, right? And our references are always European descent populations, which may or may not be similar in terms of diseases and causes to our own population. Also, that there is usually a disconnect between medical and scientific communities here. And this is just due, at least from my appreciation, to the fact that people here have many responsibilities when they are in these jobs, a lot of, for example, administrative responsibilities on top of their academic or medical responsibilities. And that means that there's very little time to really come together and discuss common goals, as it's not the case in other countries, at least from my experience. And in many cases, it's more expensive to carry our research here because we have to import many of our reagents, and that is more taxes and more like transportation expenses. So in the end, it ends up being more expensive. However, that is just to say that much of the work that we do in the lab is also to establish the facilities to do this easier, to establish the workflows, and also to train people to be able to do this work and hopefully get up to speed to, as fast as our colleagues in other countries do it. We're doing some progress there. Okay, so that was my introduction. Now I'd like to tell you a little bit about what we're doing here in Mexico to study this disease. So what we started doing was to invite patients in collaboration with the National Institute of Cancer, or Instituto Nacional de Cancerología, here in Mexico City, because they see these patients regularly. It's a reference center for these types of cancer. So basically what happened is that when these patients went to their follow-up appointments to the clinic, then the medical doctors there asked the patients if they would like to participate in our study. And if they accepted, they have to sign a consent informed sheet, and they donate a sample of their tumor. You see here a picture of that. Usually when there's a surgery for a tumor, for example, a little bit of the tumor is preserved in this state, which is called a paraffin embedded tissue. And here's the tumor, for example, and it's put in something that looks like a candle, as you can see here, but you can perfectly get DNA and RNA from here. And these are just stored at the hospital. So if the patient gives us permission to access this tissue, then we can study their DNA. And they also donate a saliva sample. And the reason for this is that in cancer studies, in order to understand the new mutations and the genes that may be important for cancer growth, we need to compare the genome that the person already carries from birth, which is, the normal set of genes inherited from the mother and the father, and compare that to the genome of the cancer, because the cancer will have already accumulated many mutations, for example, from sun exposure, from smoking, from many different life choices, for example, and exposures that the person has had. So if we compare these two genomes, we can identify the new mutations that have aroused in the tumor. And we can see by studying many tumors where these mutations tend to be. There will be some places where they are repeated, some genes that are hit harder, and that can give us hints as to what is helping that cancer grow. So that is why we need both a sample of saliva, which constitutes the DNA that the person has from birth, and a sample of the tumor. And then what we do is compare these two. That's the idea behind it. After they donate the samples, then we proceed to do DNA and RNA extraction, both from the saliva and these little candles, these tissues here. And then we send for sequencing. If you haven't heard what sequencing is, basically it's taking the DNA or the RNA, which are, again, the set of expressed genes in those cells or in that tumor, and basically put the DNA or the RNA, extract it from these tissues, and then basically put them in a machine that will tell you what the sequence, literally the sequence is. A, C, G, T, T. That's what the machine gives you, basically. It's a bit harder than that, of course. You need to do a lot of quality checks. You need to do many steps in order to be sure of what you're getting. But basically, that's what sequencing means. You obtain the actual sequence from both the tumor and the normal, and that's the way you can now compare the sequences and see where the mutations are, basically by checking the differences between those sequences. Okay, so once we do all of these, I'll show you what, the results of what we can get. But first, just a little bit of the characteristics of the patients that have formed part of this study. In the end, after about 2 years, well, 3 with the pandemic, of recruiting patients, we recruited 96 patients who donated 128 samples. And that is because some patients donate more than one sample. Some patients have what is called a primary, which is where the tumor usually initially arose. But some of these patients have, for example, metastases. So the tumor has spread to other parts of the body, and they also donate that sample, or just other samples. So that's why we have more samples in patients. And here you can see some characteristics of this cohort. We have slightly more female than male patients here. The majority are diagnosed around 2017, which is when we started the study. The majority of these patients come from the center of the country, and that may be just because the National Cancer Institute is in Mexico City, and this is closer to Mexico City. The majority of the patients are diagnosed with ulcerated tumors. And this is important because ulceration, having really an ulcer in your skin, in the tumor, is the most important prognostic factor for this population. If you have ulcerated tumors, then the prognosis is worse. That's a bad sign if you get. And as you can see, about 2/3 of the patients already have ulcerated tumors. That goes with a late stage of diagnosis, as you can see here, stage 3. And this is the same as I was telling you guys before. These patients struggle a lot to get a real diagnosis at the beginning of their journey, because these tumors just look like other diseases. So in the end, by the time they finally get a diagnosis, it's late, and they are usually ulcerated, and they are about stage 3. Here's the age. You can see there's a median of about 62 years of age where these patients are diagnosed, but you can have patients all the way from like about 30 years of age to nearly 100. So you can see there's a lot of ages where these tumors can be diagnosed. These are tumor invasiveness measurements, and this is not really that important. It's just to mention that these tumors are thick. These measurements mean that they have already, let's say, invaded deeper layers of the skin, and that is because they are late tumors. Again, this all makes sense. And finally, here in this diagram, you can see that the majority of the tumors were diagnosed in the feet. So you can see in foot soles, we have 48 of these primaries and 21 in the heels, some in the hands, and in blue, you can see the metastases, some to the central nervous system, the lungs, the liver, and bones, for example. Okay, so what can we do now when we have the sequences of the tumors and the normals and we compare them? We can get a graph like this. This work is done by Patti Basurto here, who is a PhD student in our lab. And this graph that you're seeing here is called an oncoplot. What you see here in each row is a different gene. These are names of genes. NRAS, KIT, BRAF, NF1. I mentioned these ones before, if you remember in the introduction. NRAS, BRAF, NF1. These are the genes, and then each column is a different tumor, right? And if you see a colored square, for example, that means that this tumor here, the first tumor, has mutations in NRAS, in KIT, for example, and NF1. And now the color of the squares mean the different types of mutations, because you can have many different types of mutations. You can have a change in amino acid in the protein. You can have stop codons, which means you interrupt the protein. So these are all these types of mutations that are enlisted here. And that's all to say really for this graph. At the top, you have the number of mutations in each of the tumors. And here at the bottom, you have different characteristics for these tumors. For example, here the site, if it's in the feet or in the hands or under the nails, the sex of the patient in the second one, what their age were, lighter colors mean younger patients, if the tumor was ulcerated or not, and what the tumor was, if it was a primary or a metastasis. Okay, so that's what this graph really means, and this is very standard in cancer genomics. If you do decide to go into this area, you will see this a lot. This is a typical graph, and what we can see here, and I have highlighted this here, is that in these patients, the mutational profile of acral melanoma indicates that less than 40% of these tumors have these classical driver mutations. And the classical driver mutations are the ones I mentioned to you before, that in cutaneous melanomas are about 85%. So as you can see here, there's already a big difference in the frequencies of mutations in this tumor, in these tumors. As you can see here on the right, I put a little simplified diagram of the MAP kinase pathway. This is a growth pathway, as I was saying telling you in this for the cell. And here you have sensing something in the outside, then goes to the RAS protein, and the RAF protein, and MEK, ERK, and then goes to the nucleus and transcribes a lot of genes for cell growth. So NRAS, BRAF, and NF1, as you can see here, are the first steps in this pathway. Okay, an interesting thing is that KIT is a new gene here that you see in the middle, is very highly mutated in this set of tumors. And KIT also participates in this MAP kinase pathway here by activating from a different point this cascade as well. And now if we look, this is interesting, and I'll tell you later a bit about why, but if we look now at the actual mutations that you see in these tumors, we see a plot like this. Oh, sorry, this is actually in Spanish. I didn't change that, I apologize. But basically, when you have a gene that if you mutate it, it will contribute to the growth of the cell, usually you see a pattern of mutation that is called a hotspot pattern. The first three genes here have this hotspot pattern. That means that only very specific sites, in this case of NRAS gene, position 12, 13, and 61 are the ones that gain mutations. And you see these mutations happening over and over again because those specific mutations activate the protein. And that means it activates the protein and activates the pathway. And that is the case for NRAS and for KIT and for BRAF, because these are oncogenes. Its activation activates the pathway. It's not the case for this one called NF1. As you see here, you actually have mutations that appear just once in every patient, and they are spread throughout the protein. But these mutations, as you can see, are different from these ones. They're different colors. And that means that these mutations basically interrupt the protein. So if you disable this protein, it doesn't matter if you disable it at the beginning or at the end, you're still destroying its function. So this is an important difference in how you see patterns of mutation arising in cancer. If you have a gene that you do not know what it does and you see these patterns of mutations, you can hypothesize if it's a gain of function or a loss of function. But in the end, all of these mutations, I was saying, have the same effect, which is activating the MAP kinase pathway. Okay, and just to say, this profile is actually similar to the one that has been reported in Australians and US individuals. However, an important distinction is, for example, the frequencies are very different. Whereas in those patients, the most mutated genes are BRAF or NF1. In our case is actually KIT, one of the most mutated, and that's not the case for these other patients. And we're right now investigating why that is. Okay, that's what I can say about the oncoplot, but you can also investigate parts of the genome that are gained or lost. And here, that's what you see in a plot like this. The red lines are gains of DNA segments and the losses are in blue. And if you put all samples together, then you also can see bits of the genome that are constantly gained. And again, these are usually oncogenes, genes that when they gain a function, help the cell grow. And these genes, for example, are important for, I don't know if you've heard of cyclins. These are cyclins, and cyclins are important for cell cycle progression. So if you have more of these, then maybe your cell cycle can progress more quickly, and that way you have more growth as well. And this is a tumor suppressor, CDKN2A, that is usually lost in melanomas. And here's just a comparison. Again, it's always, guys, if you do decide to go into science, it's always good to check that your results are in agreement with what has been published before. And if they are not, the first thing you have to ask yourself is, did I do something wrong? Because it tends to happen, and this is just, you know, sanity check and trying to put our results in, yes, in the context of what other people have done. And what we've seen is just, yes, indeed, this looks very similar to what has been published in other populations. However, we do have a couple of regions here that look quite different. And at the moment, we're investigating if this is a real difference, because these genes, for example, MYC is very important in cancer progression. And this may be a difference for these tumors when compared in European and Latin American populations. Now, another thing that you can do with these profiles of genes, gains and losses, is do a graph like this, in which you now put the two things I told you about before together. And basically here what you're seeing is each column, again, is a different tumor. And at the top, you're seeing which mutation it has. So if it has a BRAF mutation or an NRAS or an F1 or a KIT. And here is all the genome, chromosome 1, P arm of the chromosome 1, Q arm of the chromosome 1. And here, what you see, if in blue, there's been losses of that bit of the chromosome, or in red, there has been gains. And if you quantify this, then you see that actually 24% of the tumors have a duplication, a whole genome duplication, which is interesting. That's not very common in the other types of melanoma, for example. And also that tumors that have mutations in NRAS and BRAF have way fewer copy number alterations that tumors that have mutations in these other genes, KIT and NF1. And this is interesting because we start seeing maybe different paths to tumorigenesis. Either you gain a mutation in these BRAF or NRAS genes, they're very powerful mutations, and maybe you need less alterations in the rest of the genome to form a tumor. Whereas if you don't have such a strong driver, then maybe you need to have more mutations and more alterations to form a tumor. And maybe that's what we're seeing in a graph like this. Now, I'd like to tell you just a little bit about the RNA, because if you remember, I also told you that we extracted RNA. And here what we did is what is called unsupervised consensus clustering. And that just means you take the global gene expression of these tumors and you try to group them together just to see if they form natural groups that, groups of tumors that look similar to each other. And this is work done by Steph Vasquez, who is another PhD student in our lab. And basically what she sees is 3 different groups of tumors that you see here divided by these lines. Here at the top, I am showing you their different stages, if they have mutations in these genes that I told you before, if they have a whole genome duplication, if they're ulcerated, and what their stage is. What we think is interesting here is that this looks similar to other reports in cutaneous melanoma, in which studies like this have found, for example, 4 different types of tumors according to gene expression. And if we compare what other people have found to what we found, then we see that this first cluster is actually what we call a a mixed cluster because it expresses genes that have both immune function. They have, many different types of innate immune and adaptive immune genes expressed, but also genes that are associated with invasion. So for example, angiogenesis, which is creating new blood vessels. So this is interesting. We did not expect to see that, but that's what this cluster looks like. Cluster 1. Cluster 2, we have named proliferative, such as this one, because it expresses a lot of genes that are important for cell cycle progression and tumor growth. So we think these tumors are proliferative. And then the last group, number 3, we have named normal-like, such as this one here, because it expresses a lot of genes that are related to epidermal cell function or skin development. These are just more of a skin normal functions. And if you look at these genes here, KIT and NF1 and whole genome duplication, then you see that they tend to actually cluster with the proliferative subgroup here, whereas the best stages, so almost no ulceration and very early stages, are concentrated in the mixed cluster. Now, this is important because these characteristics can actually mark survival characteristics, and this is important for us. This is now useful in the clinic, which is what we definitely wanted to see. So this is again the paper I showed you before, and this is in cutaneous melanomas, again not acrals, but what these people found was that this proliferative cluster actually had the worst survival here, and we see actually something very similar. This is work done by Denis Serrato, who's at INCAN. And here we see also that the proliferative, the people that have been diagnosed with proliferative tumors, just according to gene expression, also have worse survival than people that have diagnosed with normal-like or mixed tumors. And also, not only there's a higher probability of, a lower probability of survival, but also there's a higher probability of recurrences, which means that the tumor appears again after it's been extracted. So we are very excited about these results because this means that by studying the genome and the transcriptome, then we can actually identify groups of patients that have differential survival. And this is now, we hope, in the future, because obviously we need to do much more studies, but this could be translatable to the clinic because now we have found markers for gene expression for tumors that are probably worse and maybe predict a worse survival than other subtypes. So this is what we're doing right now, and this is what our lab is focusing at the moment. So I have now finished this talk. I'd like just to give you a summary of what I've told you before. Before and today in this talk. So first, I told you that acral melanoma is the most common type of melanoma in Mexico and in other countries in Latin America, Africa, and Asia. And I also told you that it has been poorly studied, and that's also why it's a motivation for us to try and understand more about this disease. We have this project of acral melanoma in Latin America to elucidate the genomic profile of this disease in Latin American patients and hopefully contribute to our knowledge about this disease and close the gap in knowledge among populations. Our initial results, as I showed you today, show a similar genomic profile to acral melanoma from patients of other populations, but maybe we have some potential differences that we're investigating. We also see that genomic and transcriptomic features can identify patients with different survival. And we're doing other work. We're also analyzing the proteome of these tumors to try and corroborate observations from transcriptomic analysis. And we're also investigating the immune landscape of these tumors. Now, sorry, I do have a couple of more slides because I forgot that I also want to tell you about the collection of tumor models in Latin American patients. And I hope you will find this interesting as well. So what do we do here? So this is work that we're doing together with a researcher in Brazil called Patricia Posic. And basically what we do is we have a cohort of patients that want to participate from both Brazil and Mexico. We collect surgical samples from their tumors, and then you get these samples and then you actually inoculate them either in a mouse, and that is what is called an in vivo PDX. PDX means patient-derived xenograft. So it's a xenograft that is derived from a patient and inoculated into a mice. Or in vitro, so we try to derive cell lines and organoids from these tumors. And once they're growing, actually, we try to characterize them also by sequencing and by, well, sequencing both the genome and the transcriptome, so gene expression as well. And we hope that with these, we can then do a functional characterization of target genes. And the advantage of a model like this is that you can directly test drugs. So you have a hypothesis that this tumor would respond because it expresses a certain thing, then you can directly test a drug in it and see if the tumor responds. So just some advances here. There are 96 patients so far in the cohort, 64 from Brazil and 32 from Mexico. That equates to 110 samples. And basically, these are the characteristics here. I'm talking right now just of the Brazilian cohort because the Mexican cohort is still being built and the Brazilian cohort is more advanced. So I can tell you a little bit more about that cohort. It looks similar to what I showed you before for the Mexican cohort. There's slightly more females than males, also diagnosed between 60 and 80 years of age, also at stage 3. And most of the tumors are also from the feet, basically plantar. And many of them are metastases to the lymph nodes, and also many of them have had no treatment when their sample was taken. When you take these tumors and actually inoculate them in mice, then you see that about half of them grow, so that's the green bit here, and 31 just didn't grow. You can just inoculate a tumor into a mice, and for many reasons, it just doesn't grow. And there are also can be some adverse effects where the mouse, for example, gets infected by another thing or just dies for other reasons, and some others that are in progress in the tumor rate. So that means about a 56% success rate for growing these tumors. Now, what predicts that a tumor will grow in a mouse? So, for example, here we can see that of the tumors that have grown, half of them are primaries and half of them are metastases, but we inoculated many more primaries than metastases. So that means that actually being a metastasis is much more likely to grow because it's a more advanced tumor by definition. And also, this is again the measurement for thickness. If you have a thicker tumor when you extract, then it's more likely that it will grow, again, because it's a more advanced tumor. And just to say that these models actually recapitulate the histological characteristics in the tumors. And as here you can see, for example, what the histology of the patients look like, and this is what the tumors in the mice look like. And these are very similar at the histopathological level. And also they can also have metastatic spread, just like in the patients. And about half of them, a little bit less than half of them, actually metastasize also to the lungs and the brains of the mice. So in many cases, they behave like the human tumors. Okay, so I've finished with this bit now. And just to tell you that the ongoing work related to these is that we are now doing the genomic characterization of these tumors. We know now that 10% of these samples have mutations in BRAF, but we're still studying the rest. We're also characterizing the RNA, the transcriptome level, to find genes, for example, involving metastatic spread, or that are infusions that are both in patients and PD examples, which just means that they are important for tumor growth. Also, of course, in each of these models, tumor cell spread is monitored in the lungs, in the brain, and the liver. Patricia's team has been able to derive 6 acral melanoma cell lines, and this is actually quite hard for these tumors. So this is, 6 acral melanoma cell lines is a lot, and that allows us to do more functional studies in them, which we're very excited about. And there's also discussions to try and attempt to model anatomical location, because these tumors usually grow in hands and feet, and the back of the mouse, which is where you usually inoculate, is not very similar to the skin in the hands or the feet. So there's discussion to try and also model that by inoculating the tumor in the foot soles. And Here in Mexico, we have now inoculated 7 samples, and one of them shows a growth, and the other ones are still under observation. Okay, so a summary of this bit is that we are working to establish this patient-derived xenographic collection from Latin American patients, that most inoculated tumors are stage 3, they are treatment naive, and from older patients. At the moment, 40 PDX models have been established with a 66% success rate. It is more successful when done for metastases and thicker tumors. They also recapitulate histological characteristics seen in patient tumors. Half of these show metastatic potential, and ongoing work is focused on the genomic and transcriptomic characterization of this collection. And now I have finished, so just a slide for a take-home message is that we hope that with this work, we can contribute to the efforts to molecularly characterize this disease and hopefully to close the gap in knowledge from understudied populations such as Latin American populations. And finally, the acknowledgements. There's a lot of people that have participated in these studies. So thanks to the lab, to the patients, of course, that have participated, to our collaborators, and to you for listening. And I'm happy to take any questions. Thank you very much.
Questions, guys? Yes? Try it that way.
So, wait. Yeah. Jose Luis Peregrino here. I was wondering what's the reason for it to be most common in the feet or in the hand?
This is an excellent question, because we don't know. So, okay, we have some hypotheses, but we actually do not know. So, for example, when you look at where you hear me, right? It's just that I hear some echo. You hear me? Yeah. Yeah. Okay. When you look at, if you look at the map of the foot where the tumors actually arise, they always arise in the pressure zones of the feet. So, for example, we have this, the foot where there's a little bit around here where it doesn't touch the ground usually. So those parts of the feet actually almost don't have any tumors. Most of the tumors are in the heels, as I was showing before, or actually around, where the foot actually stands. So a hypothesis as to why it is more common in the feet is due to that, to pressure, mechanical pressure. And we are in collaboration now with a group in New York who are helping us try and test this hypothesis by mechanically actually squeeze acral cells, and they actually see some differences molecularly in what happens when you do that, right? So and yes, there are differences. Whether they're related to cancer or not, we don't know yet, but it could be something related to that. People have studied, another reason why that may be the case is because there's another study where people that are obese actually were studied that have acral melanoma. And the reason why it's important that these people are obese is because they studied their feet, and their feet actually are like, just all of the feet touches the ground, let's say. And those people have tumors all around their feet, right? So it seems that whether you're in touch with the floor or not matters, and that may be just more important for the feet than the hands. So yeah, I mean, that may be the reason. Is maybe a mechanical pressure issue, but we don't know. It's a great question that we're also trying to investigate. Thanks for that.
Can you hear me, Dr. Robles? Yeah, great. So first, thank you for that wonderful talk. I learned a lot, and I thought, it's amazing that you guys are doing all this work over here, which is so urgently needed. And one question I'm left with is whether you consider doing some work to associate these profiles, the transcriptomic profiles you're seeing, with whether the patients responded to treatment or not, and which subtypes of treatment, including if any of them were treated with immunotherapy agents?
Yes. No, absolutely. Another great question. So yes, the answer is yes. So what we're at the moment, we cannot really do that right now for two reasons with this cohort. The first one is it's a newer cohort. It's recently collected, which means that many of these patients are still undergoing treatment. We don't really know what the response rate is, and many of them have just gone to chemotherapy. Half of them actually have had no treatment, and only 3 have gone through immunotherapy. So with the data that we have now for these patients, we wouldn't be able to exactly do that. But what we want to do, and is exactly to this question, is we're trying to do drug screens in collaboration with some researchers in Florida, in Tampa, where with these cell lines that I said that were derived from the Brazilian patients and the ones that we're trying to derive here from Mexican patients, you can do drug screens. You can identify, you can say, okay, these these cell lines come from tumors that have this transcriptional profile or genomic profile. And then we can just do a massive screen. At the moment, these 300 drugs that are either FDA approved or in process of being FDA approved. And then you see, this is just a cell line thing at the beginning, but you can find candidates of potentially, oh, this profile maybe is related to a response to this drug. And that's just a hypothesis generating tool, a massive thing. And then you can model that more specifically with more cell lines, for example, or with a mouse, and hopefully give a hypothesis exactly as you're saying, okay, we see that these specific, for example, cluster 2 people or tumors respond to this drug, right? So maybe there's a basis to suggest some sort of a clinical trial with more in the future, with more studies, or genomic and transcriptomic put together, which is also what we're trying to do. So exactly right. That's what we want to get to in the end, and that's the utility of the animal models that we're saying now, because you can test the drugs in them and see if the tumors respond or not, right? So yes, that's where we want to get to with all of these studies.
Curious question: have you guys tried to see, or is that data available? Because I've never seen it. Whether Hispanic patients living outside of Latin American countries also have this higher frequency of getting April as opposed to cutaneous melanoma? For example, Hispanics living in the US.
Can you repeat just the first bit, because I didn't get it, sorry.
Whether patients who are of Hispanic ascent but don't live in Latin American countries also have this higher frequency of April melanoma as opposed to cutaneous melanoma.
So they do. I'm going back to the, let me just go back a more intelligent way. Well, no, it's not going to be a more intelligent way. I'm just going to go back. Okay, here. So, so if you see here, wait, where's my mouse?
Wait, I don't have your screen up. I think you stopped sharing.
Oh, sorry.
Yeah.
Can they see them? Can they see it? Or I can just tell them.
Okay, let me switch, yeah.
Or I can tell you if it's too hard to see it again.
There, we see it. We see
it. You see it, okay. So so as you can see here, for example, we have 5 circles for the US, and that is because the US reports by what they call race. It's a very controversial term because other countries do not use this term. And for example, Hispanic is actually not a race for them. It is not a race, it's an ethnicity. And that means that people that are white, African, Asian can be Hispanic as well. So it's like an overlapping term. But this is what they reported in their massive study of the ZEER data. And they say, okay, yeah, for European descent individuals, you see a lot of green and very little red. But for the Hispanics, the people that identify themselves as Hispanics, you see more red here. And the US African Americans, you see also a lot of red here. So this can be related to several things. The first one is that European descent people tend to be lighter skinned, and that means they're just more susceptible to get UV induced melanoma. But that's why they have a lot of green as well. So that's the first most important issue, I think. But the second is that some people have actually reported, we don't know why yet, and that's also something that we're studying, the relationship of genetic ancestry with specific mutations, for example, or specific diseases. Some people have already reported with massive data sets that, for example, African Americans do have a higher incidence of this subtype of melanoma. We don't really know why. I mean, is it only mechanically related? If so, do they have more mechanic stress than other people? I mean, who knows? So yes, the answer is yes, but we don't, also it's because we don't know the causes. Maybe I didn't even say this, it's very important. We don't know the causes of this melanoma, it's not UV related. So we don't know why that is, but yes, we see these differences in different yes, ethnicities here, which is interesting. But as we don't know the causes, we just don't really know how to relate it to the observations, right? We're still, stay tuned, we're still on it.
We can actually go on a few more minutes if people have more questions.
I think Dr. Gonzalez had her hand.
Yes, I saw that in the Zoom. So we'll do that and then we'll go back to. So
You tell me what to do and I'll do it.
yeah, we have a question from Zoom.
You tell me when I can go.
You can go now.
Okay, thank you. Thank you very much, Dr. Robles, for this lovely talk. I've heard it twice, and I have to say that every time that I hear it, I like it more than the previous time.
You're too
So,
kind.
I didn't know about the part of the patient-derived organoids, and wow, that's great. I've worked on that as well, and having sick cell lines, that's a thing, you know, that they like to, they are a little bit picky, and but it's congratulations on that part. It's very cool that you have also considering that issue. So I have a question regarding I don't know if you were aware of this, but there was a publication last year in October 2023 by Andres Moreno Estrada, this group that they created the first Mexican biobank,
Yeah.
for medical genomics and of diverse ancestries in Latin America and stuff.
That's right.
So I wanted to ask, first of all, if these type of publications or this type of work that they do benefit or contribute to your work, and if so, how it goes.
Absolutely, and we are using the data actually. Another great question. So yes, we are collaborating with Andres. Actually, before they published their paper, they gave us the data to start and using it as well. And the way we can use it is that because for them is what they publish is really was collected by the INEGI census in like the 90s. Like, it's been, it's all across the country. And the way we can use this data is because they genotyped 6,000 of those people. And we have those genotypes and we can use them as controls for our data set. So we also genotyped our samples. I didn't show this data right now, but the way we can do that is also compare, for example, we have patients from Mexico and we can genotype them, which means is determining, for the students that haven't heard this term, means determining the actual base at different locations in the genome, but across the whole genome. And in that way, you can reconstitute, the bits of genome that are missing because genomes are, DNA is inherited in like chunks. So if you have, this base and this base, then usually you can reconstruct with high accuracy what's around it. But you don't get the sequence. It's different from sequencing. You just get a base every, certain number of bases. So they've done that, and we've done that too. And the way you can try and do here is compare again these like controls and patients, and try to see if you have more of a certain region of the genome, like more common in your patients than in your populations. And another student, I didn't present his data today, but another student is actually doing that exactly. It's a bit hard because for that we have 220 patients, I think, but it's still not enough to have, and the Mexican is very diverse. So we have, for the biobank, we have had to remove, for example, all the Yucatan people because we don't have patients from Yucatan. And, so you have to clean the data set and then compare and try to identify these changes. So, and that is to try and identify if there's genetic causes for this cancer, which we haven't found any yet, or no one has yet. But that's the way we can use the Mexican biobank, and we are at the moment also trying to just use them as controls for our sample. So yeah, great question too. Thank you for that.
Thank you.
Thank you.
Thank you. Any final question? Yes, Juan, go ahead.
Why do you think that melanoma can have so many mutations in comparison
comparison to other tumors? Like, does it have any specific characteristics that makes it so easily mutable?
Another great, you guys make great questions. Before answering this, just to say, we have had students from you guys from ASU in our lab, and they have been great. So just to let you know, if you're interested in these topics, let us know, because yeah, we'd love to hear from you. Okay, for this question, yeah, so melanomas are strange in the sense that they can accumulate a lot of mutations and don't die. That's why they don't respond too well to chemotherapy. And the reason why some people have speculated this is the case is because of their job, their function in the body. So as I said in the beginning, the function of melanocytes is to be the first line of defense against UV light. And UV light causes a lot of mutations, like, a lot. Melanomas as a general are the type of tumor that have the most number of mutations than any other tumor, if you look at the atlases. So why do they resist so much and they don't die? And you put the chemotherapy and they don't die, because they are programmed transcriptionally, like their let's say intrinsic characteristics, or the programs that they express, are tuned for survival, because they need to get a lot of mutations from the sun, like they need to detect it and start producing melanin. So they detect and produce, and they're also in the skin. So that's like super mutagenic, right? Like you have to be able to survive if you're in the skin, because you're getting everything, in your genome. So they are programmed genetically, people think, to just get anything in it and just don't die. That's why it's so hard to kill them and to treat them. And it's just thought to do, due to their function. That's the answer I can give you. That's what people have investigated. Like, oh, they express these survival modules, and they are just hard to kill. They just don't die as easily as other cells when you induce more mutations in them. But that's also why they do their function the way they do it. Like, they can produce the melanin. That's the tanning response, guys. If you go to the sun and you get tanned, that's the melanocytes being like, here's some melanin, please don't go to the sun, guys. Don't get tanned, it's bad. That means the melanocytes are stressed. They're stressed.
Very well. So we'll have to stop it there, but that was an amazing talk, and we thank you again.
Thank you so much. Thank you so much. It's been a pleasure. I hope I can visit you guys sometimes in person. Thanks so much.
And you can put up your contact information or your final slide again.
Yes, it was the first slide, but yes, absolutely.
Oh, yeah.
I can. Let me put it there. Here. So on the bottom right, I haven't updated this webpage in a while. I'm sorry, I'm terrible. But if you write to me in my email, or I am more active on Twitter, that's that's ways to contact us if you're interested.
Right. Thank you very much.
Thank you so much, everyone. Enjoy your week. Looks amazing.
Very well, guys. So we have right now the science fair. I'm told there was one team that is signed up. So that is going to be happening at the physics lab. And other than that, we come back at 2 for our first roundtable. Then we have the debate at 4, and at 5 we have a couple more games of STEM ball.